welcome back to the deep dive.
No.
Okay, first year MBBS biochemistry. It's a lot, right? Pathways everywhere.
It really is. Feels like drinking from a fire hose sometimes.
Exactly. So today, we're doing a focused revision, a real deep dive into 3 absolutely core topics. Hemoglobin, porphyrins, and how their breakdown connects to jaundice. This stuff is, the bedrock for diagnostics later on. We're aiming to really nail it down today.
That's the plan. And not just listing facts, but getting the concepts clear, the mechanisms. How does it work? We've structured this based on standard MBBS curriculum texts, focusing on what you genuinely need to understand.
Right. We'll cover HB's function, what happens when its synthesis goes wrong, like in sickle cell or thalassemia, and then that whole pathway from heme breakdown to jaundice.
Exactly. It's all connected, ready to jump in.
Let's do it. Starting with part 1, hemoglobin, the oxygen carrier. You can't really appreciate how clever this molecule is without looking at its structure first.
Yeah.
So HB. When we talk structure, what are we looking at? It's more than just a protein, right? It's got that quaternary structure.
Absolutely. We're talking about HbA1, the main adult form. It's a heterotetramer.
Heterotetramer, meaning 4 chains, but not all the same.
Exactly. Typically, 2 alpha chains and 2 beta chains. That's the alpha-beta-2 structure you see written down. Together, these make up the globin part.
Okay, the protein part, but the business end is the heme.
That's the functional core, definitely. Heme is this ring structure, a protoporphyrin with an iron atom right in the middle, and critically, it's ferrous iron, Fe2 plus L.
2 plus N.
That specific state, Fe2 plus L, is what can actually bind oxygen reversibly. And since you have 4 chains...
You get 4 heme groups, so 1 HB molecule carries 4 O2 molecules. Makes sense.
Precisely.
Now, we often hear about myoglobin in the same breath. How does that fit in? It looks kind of like one piece of HB.
Yeah, structurally, it's similar to a single subunit of hemoglobin. Myoglobin, or MEP, is mainly in muscle tissue. Think of it as the local oxygen storage tank.
Storage, not transport.
Right. It's a monomer, just 1 chain, 1 heme group. It grabs oxygen and holds onto it, ready for when the muscle suddenly needs a burst. And it's not trivial. Something like 5% of your body's iron is tied up in myoglobin.
Wow. Okay. So back to HB's main job, obviously, oxygen transport from lungs to tissues. But you mentioned it plays a role on the way back, too.
Definitely. Its primary role is that O2 delivery, no question. But once it drops off the oxygen in the tissues, it helps carry some of the CO2 back.
How much are we talking?
Around 15 to 20% of the total CO2 produced by metabolism gets transported back, bound to hemoglobin. It also plays a crucial role in buffering the blood pH, which is vital.
So a multitasker. Gas exchange, buffering. Quite the molecule.
Yeah.
Hashtag tag 1.2 oxygen transport mechanics.
Okay, so how does it bind oxygen so efficiently? This is where it gets really clever. It doesn't just bind one O2 then the next linearly. It uses cooperative binding.
Cooperative binding. I remember this being key. It means binding one makes it easier for the next one to bind.
Exactly. The first oxygen binding triggers a change in the whole molecule's shape or conformation. This change dramatically increases the affinity of the other heme sites for oxygen. It's like they're working as a team.
A team effort for rapid loading in the lungs.
Precisely. And this heme-heme interaction, this cooperativity, is what gives the oxygen dissociation curve its characteristic shape.
Ah, the ODC.
Yeah.
Not a straight line, but that S shape, right? The sigmoidal curve.
That's the one. It graphically shows oxygen saturation against the partial pressure of oxygen. Myoglobin, which doesn't have cooperativity, has a simple hyperbolic curve. It binds O2 tightly and doesn't let go easily.
Which is good for storage, but not transport.
Right. HB's sigmoid curve is perfect for transport. Steep in the middle, meaning small changes in oxygen pressure in the tissues cause a large amount of oxygen release. Flat at the top, meaning it loads up fully, even if lung oxygen levels fluctuate a bit.
And this shape change is physically real. We talk about T and R forms.
Yes, these are the 2 major conformational states. T stands for tense. That's the deoxyhemoglobin state, the shape it has when it's not carrying oxygen.
Tense, so low affinity for oxygen.
Exactly. It's stabilized by a network of ionic bonds and hydrogen bonds, salt bridges, between the subunits. This structure holds it tense and makes it harder for oxygen to bind. This is the state favored in the tissues where you want it to release oxygen.
Okay. So how does it switch?
When that first oxygen molecule manages to bind to a heme group, it physically pulls the iron atom slightly, moving it into the plane of the porphyrin ring. This little tug is enough to start breaking those salt bridges.
Wow.
Yeah, it causes a cascade. Breaking those bonds releases the tension, and the molecule clicks into the R form R for relax.
And the relaxed form is the high affinity state.
Correct. It readily binds the next oxygen molecules. The R form is stabilized by oxygen binding itself. So the transition from T to R explains that cooperative binding and the shape of the ODC. It's probably the best example of allosteric regulation we have.
Okay, pause for a moment here. Think about this. You've got a molecule that literally changes its shape, breaking internal bonds, not just to pick up cargo, but to make itself better at picking up the next pieces of cargo.
Yeah.
That's incredibly efficient design.
It really is a self-regulating molecular machine.
So this T to R shift, it's not just about oxygen binding. The conditions in the tissues actually influence it, right? Pushing it towards the T state to release O2.
Absolutely. The molecule is exquisitely sensitive to its environment. There were 4 key players, 4 allosteric effectors that signal, hey, release oxygen here. These are carbon dioxide, CO2, protons, H+, chloride ion, Cl, and a molecule called 2-PO3-bisphosphoglycerate, or 2-Port3-BPG.
Let's tackle the big ones first, H+ and CO2. This is the Bohr effect, isn't it?
Yes, the Bohr effect. Simply put, it means that hemoglobin's affinity for oxygen goes down when the pH decreases, meaning more H+ ions, or when the partial pressure of CO2 goes up.
Which happens exactly where you need it to in active tissues that are churning out acid and CO2 as metabolic waste.
Precisely.
So how does CO2 actually do that? Does it bind where oxygen binds?
Not directly to the heme iron, no. CO2 can bind to the N-terminal amino groups on the globin chains themselves. This forms carbaminohemoglobin.
Carbaminohemoglobin, okay.
And when CO2 binds there, it introduces negative charges, which help to form new salt bridges, stabilizing that low-affinity T state. This chemically encourages oxygen to pop off.
And the protons, the H+ all?
Protons do something similar. They bind to specific amino acid residues on the globin chains, particularly histidine residues. This protonation adds positive charges, again strengthening those salt bridges that hold the molecule in the T state.
So more CO2, more H+ off, stronger T state, more oxygen release. The ODC curve shifts to the right.
Exactly. A rightward shift means lower affinity at any given oxygen pressure, facilitating unloading in the tissues. It's incredibly effective.
You mentioned HB acts as a buffer too.
A very important one. It's the main non-bicarbonate buffer in blood. When protons bind to HB in the tissues, helping release oxygen, the HB effectively mops them up. The numbers are quite neat. For about every 2 protons bound, 4 oxygen molecules get released.
And it all reverses in the lungs?
Perfectly. In the lungs, high oxygen levels favor binding, which forces the release of those protons. The released protons then combine with bicarbonate, regenerating CO2, which you exhale. It's a beautiful cycle.
Okay. Now, you also listed chloride, COL, as an effector. How does that fit in?
Yeah, chloride's role is a bit less direct than CO2 or H+ or so, but it's still important. It also helps to stabilize the T state, often by interacting with positively charged groups that become available in the deoxy conformation. Think of it as another factor reinforcing that low affinity state.
Got it. And the last one, 2,3-BPG. You said it's maybe the most powerful internal modulator.
Yes, 2,3-BPGA is really significant for fine-tuning oxygen delivery. It's made right inside the red blood cell itself, through a little side chute off the main glycolysis pathway.
Okay, so the red cell makes its own regulator.
It does, and 2,3-BPG is a highly negatively charged molecule. It finds a specific binding spot right in the central cavity between the 4 subunits of hemoglobin, but crucially only when HB is in the T state.
Why only the T state?
Because the cavity is the right size and has the right positively charged residues lining it only in the T conformation. When 2,3-BPG slots in there, it forms additional salt bridges cross-linking the beta chains. This makes the T state even more stable.
So it strongly pushes HB towards releasing oxygen.
Exactly. It significantly decreases oxygen affinity. This is really important physiologically. For example, at high altitude, your body adapts by making more 2,4,3-BPG.
To help unload the limited oxygen more effectively to the tissues.
Precisely. Same thing happens in chronic hypoxia or severe anemia. It's a vital adaptation mechanism. Hashtag tag 1.4 hemoglobin derivatives. Now, we also need to be aware clinically that HB can be altered into forms that don't work properly. These are the hemoglobin derivatives.
Right, like methemoglobin. What's happening there? Is it an oxygen binding issue?
It's an iron issue. In methemoglobin, the ferrous iron, Fe2+, in the heme group gets oxidized to the ferric state, Fe3+.
F3+, and that can't bind oxygen.
Correct. Ferric iron cannot bind oxygen. Now, the body has enzyme systems like NADH cytochrome by 5 reductase, to constantly reduce any F3 plus back to F2 plus re. But if these systems are overwhelmed, perhaps by certain drugs or toxins, or if there's a genetic defect in the enzyme.
Methemoglobin builds up.
Exactly. And it causes cyanosis, a bluish tint to the skin, because the MET-HB can't carry oxygen, leading to what's called a functional anemia. The total HB might be normal, but a chunk of it is useless.
Okay, that's MET-HB. Then there's the really dangerous one, carboxyhemoglobin.
Ah, yes, COHB. This forms when carbon monoxide, CO, binds to the heme iron.
With CO, like from exhaust fumes.
Exactly. It's a major air pollutant, odorless, colorless, very dangerous. The problem is CO binds to the same site as oxygen on the heme iron.
But it binds much more tightly.
Way, way more tightly. Its affinity for hemoglobin is about 200 times higher than oxygen's affinity.
200 times.
Yeah, so even very low concentrations of CO in the air can tie up a significant percentage of your hemoglobin, preventing oxygen from binding. It effectively suffocates you from the inside.
And I remember reading it has another nasty effect, too.
It does. When CO binds to one heme site, it locks that subunit and tends to lock the other subunits on the same molecule into the high-affinity R state.
So even the oxygen that is bound can't get released properly in the tissues.
Exactly. It shifts the ODC curve way to the left and makes it more hyperbolic.
Yeah.
So you get reduced oxygen carrying capacity and impaired oxygen unloading. That's why CO poisoning is so lethal.
Okay, so we've seen how HB works beautifully when everything's right. Now, let's transition into what happens when the genetic instructions for making it are flawed. This brings us to hemoglobinopathies and Thalassemias.
Right. And before we dive into specific diseases, it's worth remembering that the balance of synthesis is critical.
You mean making the heme part and the globin part?
Exactly. Heme synthesis and globin chain synthesis have to be perfectly coordinated. The cell needs the right amounts of alpha chains, beta chains, and heme all at the same time. In fact, free heme itself acts as a signal
to stimulate globin production.
Yeah, it helps regulate the process. If you make way too much of one component or not enough of another, the whole system breaks down and the red cells suffer.
Okay, so the classic example of things going wrong is sickle cell anemia, HBS. That's a problem with the globin chain itself, right? A mutation.
It is. Sickle cell anemia is the textbook example of a hemoglobinopathy, an abnormality in the structure of the globin chain. It's caused by a single point mutation in the gene for the beta globin chain.
Just one change in the DNA.
One single base change. Adenine replaces thymine, T to A, in the DNA code. This changes the mRNA codon from JAG to GUG.
And that GUG codon codes for a different amino acid.
Yes. Instead of glutamic acid, which is normally at the 6th position of the beta chain, you get valine.
Glutamate to valine at position beta 6. That sounds like a small change, but obviously it isn't. Glutamate is charged, valine is hydrophobic, right?
Exactly. You swap a hydrophilic, water-loving amino acid for a hydrophobic, water-hating one right on the surface of the protein. And this has massive consequences, but only under certain conditions.
What conditions?
Only when the hemoglobin gives up its oxygen when it's in the deoxy, tense state.
The T state again?
Right. In the T state, the shape change exposes a little hydrophobic sticky patch on the surface of the HBS molecule, and this new valine at position 6 on another HBS molecule fits perfectly into that patch.
So they stick together.
They stick together. When oxygen levels drop, like in the capillaries, the deoxy HBS molecules start linking up, polymerizing into long, rigid fibers inside the red blood cell.
And those fibers distort the cell into the sickle shape.
Precisely. That's where the name comes from. These sickled cells have huge problems. They're stiff. They can't squeeze through narrow capillaries.
Leading to blockages.
Yes, vasoocclusion. This cuts off blood supply to tissues, causing severe pain, the sickle cell crises, and organ damage over time. Plus, the sickled cells are fragile and break apart easily, leading to chronic hemolytic anemia.
It's a devastating disease for people who have 2 copies of the mutated gene. Homozygous.
It is. But interestingly, people with just one copy, the sickle cell trait, heterozygous, have a mix of normal HbA and HbS. They're usually perfectly healthy.
And they actually have an advantage in some parts of the world.
Yes, the sickle cell trait provides significant protection against severe malaria. The malaria parasite has trouble thriving in red cells containing HbS. It's a fascinating example of natural selection in action.
How do we typically detect HbS in the lab?
The classic method is hemoglobin electrophoresis. Because Vine has a different charge than glutamate, HbS moves differently in an electric field compared to normal HbA. You can see the different bands clearly.
Okay. Now, the sources mentioned other hemoglobinopathies too, like HBC, D, and E. Are they similar mutations?
They're also point mutations leading to amino acid substitutions, often in the beta chain. For example, HBC disease involves a change at the same position, beta 6, but this time glutamate is replaced by lysine.
Lysine, that's positively charged.
Right, so again, a change in charge and structure at a critical spot, leading to a different set of problems, usually milder than sickle cell, but still causing anemia. HBD and HBE involve different substitutions at other locations. Hashtag tag 2.2 Thalassemias. Okay, so if hemoglobinopathies like sickle cell are about the quality of the hemoglobin and a faulty structure, then thalassemias are about the quantity.
Quantity, meaning not enough is made.
Exactly. Thalassemias are a group of inherited disorders where there's a reduced rate of synthesis or even complete absence of one of the globin chains, either the alpha chain or the beta chain.
So it's an imbalance. If you don't make enough alpha chains, you have leftover beta chains and vice versa.
That's the core problem. Let's say you have beta thalassemia, where you can't make enough beta chains. The alpha chains are still being produced normally. These excess unpaired alpha chains are unstable.
What do?
They precipitate inside the developing red blood cells in the bone marrow. This damages the cells, leading to their destruction before they even mature. That's called ineffective erythropoiesis. The few cells that do make it into circulation are also abnormal and get destroyed quickly, hemolysis. The result is severe anemia.
Okay, so we classify them based on which chain is affected, alpha and beta thalassemia.
Correct. Let's start with alpha thalassemia. This involves problems with making the alpha chain. Now, humans normally have 4 genes coding for the alpha chain, 2 on each chromosome 16.
4 genes, so the severity depends on how many are faulty or missing.
Exactly. If 1 or 2 are gone, you might have mild anemia or be just the silent carrier. If 3 are gone, you have a more significant condition called hemoglobin H disease. But if all 4 alpha genes are deleted,
This sounds really bad.
it's incompatible with life. This causes hydrops fetalis. The fetus can't make any functional hemoglobin like HbA or fetal HbF, which both need alpha chains. They develop severe anemia, massive fluid accumulation, edema, and usually die before or shortly after birth. Devastating. Okay, what about beta thalassemia?
Beta thalassemia results from deficient synthesis of the beta chain. We normally have 2 beta globin genes, 1 on each chromosome 11. Again, the severity varies.
Beta thalassemia minor versus major.
Right. Beta thalassemia minor trait usually involves 1 faulty gene and causes mild anemia. But beta thalassemia major, also known as Cooley's anemia, occurs when both genes are severely affected. Beta chain production is minimal or absent.
And the consequences.
Severe life-threatening anemia starting in infancy. The excess alpha chains cause massive red cell destruction. Without lifelong blood transfusions, these children typically died within the first few years. Even with transfusions, iron overload becomes a major problem.
You mentioned earlier that the cause isn't always just a simple mutation changing an amino acid. There was something about RNA processing. Yes, that's a really important point, especially for understanding beta thalassemia, where the genetic causes are very diverse. Some cases are due to deletions or nonsense mutations stopping protein production, but many are due to mutations that affect gene regulation or RNA processing.
Like splicing.
Exactly. A single nucleotide change near an exon-intron junction can completely mess up how the pre-messenger RNA, hnRNA, is spliced together.
So the introns don't get removed correctly.
Right, or an exon gets skipped, or a cryptic splice site within an intron gets activated. The end result is an abnormal mRNA molecule that either can't be translated properly or produces a useless protein fragment. So no functional beta globin is made, even though the main coding sequence might look okay. It highlights how errors in processing can be just as bad as errors in the code itself.
Fascinating. Okay, we've covered a lot on HB problems. Quick recap question for everyone listening. Can you recall that single amino acid substitution in HBS? Where does it happen and what's the change?
Give yourself a second. It's glutamate change to the valine at position 6 of the beta globin chain. Remembering that glu-valley-val beta-6 connection is absolutely key.
And maybe a memory trick for those 4 allosteric effectors that push oxygen off HB.
Let's see. You could think of active tissues being charged with waste, CO2, acid, H+, and they need help, so BPG steps in, and maybe Cl tags along. It's a bit stretched, but CO2, H+, BPG, and Cl are the 4.
All right, let's shift gears now. We've talked a lot about the globin part and the whole HB molecule. Now let's zoom in on that functional core, the heme group itself and how it's made. This involves porphyrins.
Right. Porphyrins are these fascinating, intensely colored cyclic molecules. Their basic building block is a smaller ring structure called a pyrrole.
Pyrrole.
4 chiral rings get linked together, usually by little carbon bridges called methanol bridges, to form the larger porphyrin ring. This tetrapyrrole structure is absolutely fundamental. It's the basis of heme in animals, but also chlorophyll in plants.
So same basic ring structure, doing very different jobs.
Exactly. And while we're focused on heme synthesis, the source material does briefly mention another interesting application: porphyrins in cancer therapy.
Really? How does that work?
It seems some porphyrins tend to accumulate in tumor cells. When you then expose them to specific wavelengths of light, they become activated and generate reactive oxygen species that can kill the cancer cells. It's called photodynamic therapy.
Interesting side note. But okay, back to our main path, making heme for hemoglobin. This biosynthesis pathway seems complicated, and errors here cause the porphyrias, right?
They do. Understanding this pathway is crucial because defects at different steps lead to very different and sometimes very severe diseases. Hashtag tag 3.2 biosynthesis of heme, the pathway and regulation.
Okay, heme synthesis. It involves multiple steps, bouncing between different parts of the cell.
Yeah, it's an 8-step pathway that starts in the mitochondria, moves out into the cytosol for a few steps, and then finishes back in the mitochondria.
Let's hit the highlights. Where does it start? What are the raw materials?
It kicks off in the mitochondria. The starting materials are succinyl-CoA.
From the TCA cycle, right?
Exactly, from the Krebs cycle, and the amino acid glycine. These two molecules condense together.
And the enzyme for this first step is the crucial one, the rate-limiting step.
Absolutely. This first reaction is catalyzed by ALA synthase. That's alpha-aminolevulinate synthase. This is the key regulatory point for the entire pathway, especially in the liver.
Okay, ALA synthase, what does it make?
It forms an unstable intermediate alpha-aminobeta ketoadipate, which quickly loses a carbon dioxide molecule to become ALA, or alpha-aminolevulinate.
ALA.
Got it.
Then ALA leaves the mitochondria.
Yes, ALA moves out into the cytosol. The next really important step happens there. 2 molecules of ALA combine to form a pyrrole ring called porphobilinogen, or PBG.
Okay, so we've made the basic pyrrole unit now.
Right. Then there's a series of enzyme-catalyzed steps in the cytosol, where 4 PBG molecules are linked and modified to build up the larger porphyrin structure. Eventually, this leads to a molecule called protoporphyrin IX.
Protoporphyrin IX. Is that the final ring?
It's the final porphyrin ring structure, yes, but it's missing the crucial ingredient.
The iron.
Exactly. For the very last step, the protoporphyrin IX moves back into the mitochondria. There, the enzyme for chelatase, sometimes called heme synthetase, inserts a ferrous iron atom, Fe2+, right into the center of the ring.
And boom, you have heme.
That's heme.
Now, you mentioned for chelatase. I remember something clinically important about that enzyme. It's sensitive to
Lead. Heavy metals, but especially lead, Pb2+.
lead poisoning.
Yes. Lead strongly inhibits ferrotelatase. It blocks that final step of inserting iron into the porphyrin ring. This is a major reason why lead poisoning causes anemia. You simply can't make enough heme. Lead also inhibits an earlier enzyme in the pathway, ALA dehydratase, compounding the problem.
Okay, that's a critical clinical link. Now, how is this whole pathway regulated? You mentioned ALA synthase is key.
Regulation is a bit different depending on the cell type. In erythroid cells, the ones making hemoglobin in the bone marrow, the main goal is to make huge amounts of heme to match globin production. So regulation is partly on later enzymes like ferrotelatase, and importantly, the heme produced feeds back positively to stimulate globin synthesis. Keep it balanced.
Makes sense. What about other cells, like in the liver, that just need heme for enzymes like cytochromes?
In non-erythroid cells like the liver, the regulation is all about preventing overproduction. It's classic negative feedback focused entirely on that first enzyme, ALA synthase.
So the final product, heme, inhibits the first step.
Exactly. Free, unbound heme acts as a potent inhibitor. It works in several ways. It directly inhibits the activity of ALA synthase. It suppresses the synthesis of the ALA synthase enzyme by reducing gene transcription and translation. And it even blocks the transport of the newly made enzyme into the mitochondria where it needs to function.
Wow, multiple layers of control.
It has to be tightly controlled because the intermediates in the pathway can be toxic if they accumulate.
And this regulation ties into certain drugs causing problems, right? Something about P450 enzymes.
Yes, absolutely. This is crucial for understanding why some drugs can trigger attacks of porphyria.
Oh.
Many drugs, like barbiturates, for example, phenobarbital, or certain anticonvulsants, as well as alcohol and some environmental toxins, are metabolized by the cytochrome P450 system in the liver.
And cytochrome P450 enzymes contain heme.
They do. They're hemoproteins. So when you take one of these inducing drugs, the liver ramps up production of P450 enzymes to handle the detoxification load.
Which uses up a lot of the liver's free heme pool.
Exactly. The heme gets consumed rapidly to make all this new P450. This drop in the free heme level removes the negative feedback on ALA synthase.
So the brake comes off ALA synthase.
Precisely. The gene gets strongly derepressed, the liver starts churning out huge amounts of ALA synthase, and the whole heme synthesis pathway goes into overdrive up to the point of the inducing agent. If someone has an underlying genetic defect further down the pathway,
the intermediate just before the block will build up massively, triggering an attack.
That's exactly what happens in acute porphyrias, hashtag 3.3 porphyrias, clinical diseases of heme synthesis. So the porphyrias are this group of mostly inherited diseases where there's a defect in one of the 8 enzymes of heme synthesis. Which disease you get and what the symptoms are depends entirely on which enzyme is deficient.
And where the intermediates build up, because you said some are toxic.
Right. The accumulating intermediates are the problem. Broadly, you can think about 2 main types of symptoms, neurological problems and skin problems, photosensitivity.
Okay. Which ones cause which?
The earlier intermediates, specifically ALA and PBG, seem to be primarily neurotoxic. The classic example is acute intermittent porphyria, or AIP.
AIP. What's the defect there?
It's a deficiency in the third enzyme, hydroxymethylbilane synthase. You might also see it called PBG diminis. Because this enzyme is blocked, the precursors ALA and PBG build up significantly.
And these cause the neurological symptoms?
Yes. Patients get attacks of severe abdominal pain, nausea, vomiting, psychiatric symptoms like anxiety or confusion, and peripheral neuropathy, weakness, or pain in the limbs. But importantly, AIP typically does not cause photosensitivity. Their urine might turn dark on standing, though, due to PBG polymerizing.
Okay, so AIP equaled neurovisceral symptoms, high ALA and PBG. No skin issues. What are the ones with skin issues?
The photosensitivity happens when the porphyrinogen intermediates the ones that are already tetrachiral rings accumulate. These molecules can absorb sunlight energy.
And that causes damage.
Yes. When they absorb light, they get into an excited state and transfer that energy to oxygen, creating reactive oxygen species like singlet oxygen right there in the skin. This damages skin cells, causing blistering, fragility, pain, and scarring on sun-exposed areas.
Which porphyria is the classic example of this?
Congenital erythropoietic porphyria, CEP, also known as Gunther's disease, is a severe example. It's due to a defect in uroporphyrinogen, the third synthase. This leads to massive accumulation of uroporphyrinogen first isomers.
And severe skin problems.
Extremely severe photosensitivity from early childhood, often leading to disfigurement. Their urine is often reddish, and their teeth might fluoresce red under UV light because the porphyrins deposit there too. The source even mentions the liver showing fluorescence.
Wow. Are there others that mix symptoms?
Yes. For instance, hereditary coproporphyria, HCP, involves a defect in coproporphyrinogen oxidase. Patients accumulate coproporphyrinogen the third, but also ALA and PBG can build up, so they can have both photosensitivity and acute neurological attacks similar to AIP. Variegate porphyria, VP, is another mixed type.
And the most common type overall.
That's porphyria cutanea tarda, PCT. This one is usually acquired later in life, often triggered by liver damage, like from alcohol, hepatitis C, or iron overload, although there's often an underlying genetic susceptibility involving the enzyme uroporphyrinogen decarboxylase. PCT mainly causes blistering photosensitivity, but not usually the acute neurological attacks.
Okay, so a whole spectrum of diseases depending on the enzyme defect, and some can be acquired or triggered.
Yes, besides PCT, there are also purely acquired or toxic porphyrias, most notably from lead poisoning, which we already discussed inhibits furochelatase and ALA dehydratase.
Okay, we've built the heme, we've used it in hemoglobin, but red blood cells don't live forever, right? About 120 days.
Roughly, yes. After about 4 months, the older, less flexible red cells are removed from circulation, mainly by macrophages in the spleen and liver, the reticuloendothelial system.
And the hemoglobin inside gets broken down. What happens to the heme part? It can't just be thrown out, can it? Iron is precious.
The iron is definitely recycled. The globin chains are broken down into amino acids, which are reused. The heme ring itself, though, that gets dismantled. This is the start of the pathway leading to bilirubin and bile pigments.
Okay, take us through it. How do we get from heme to bilirubin?
It starts with an enzyme called heme oxygenase. This enzyme breaks open the porphyrin ring, releasing the iron, which gets picked up by transferrin, and also releasing carbon monoxide, interestingly.
CO. The body makes its own CO.
Tiny amounts, yes, during heme breakdown. The opened ring structure is now a linear molecule called biliverdin. Biliverdin is green. It's one of the pigments you see in bruises sometimes.
Ah, okay. Green biliverdin. Then what?
Biliverdin is then quickly reduced by another enzyme, biliverdin reductase, to form bilirubin. Bilirubin is a yellow-orange pigment.
So hemoglobin green,
Yeah.
bilirubin yellow.
Exactly. Now, this bilirubin, formed initially in the spleen and other places, is very hydrophobic. It's not soluble in water or blood plasma at all.
So it needs a chaperone to travel.
It does. This is unconjugated bilirubin. To get to the liver for processing, it binds tightly to albumin, the main protein in blood plasma. Albumin acts as its transport vehicle.
Why is it so important that it binds to albumin?
Because free, unconjugated bilirubin is lipophilic. It can cross cell membranes, including the blood-brain barrier, especially in newborns whose barrier isn't fully developed. If levels get too high, it can deposit in the brain and cause serious neurological damage called kernicterus. Binding to albumin keeps it safely contained in the bloodstream.
Okay, so albumin carries the unconjugated bilirubin to the liver. What happens there? This is the conjugation step.
Yes, the liver's job is to take this insoluble, potentially toxic bilirubin and make it water-soluble so it can be excreted. It does this by conjugating it, attaching molecules of glucuronic acid to it.
Glucuronic acid, where does that come from?
It's provided by a carrier molecule called UDP glucuronate. The enzyme that does the attaching is UDP glucuronal transferase, often abbreviated UGT. Usually 2 glucuronate molecules are added to each bilirubin molecule, forming bilirubin diglucuronide.
And this conjugated bilirubin is water-soluble.
Yes, highly water-soluble and non-toxic. It's ready for excretion. And it's worth noting that same UDP glucuronate system is also used by the liver to conjugate and detoxify many drugs and other waste products. It's a central detoxification pathway.
Okay, so now we have water-soluble conjugated bilirubin in the liver. How does it get out?
It's actively transported, pumped out of the liver cells into the bile canaliculi, the tiny channels that collect bile. This transport step is energy dependent and can be a bottleneck. The conjugated bilirubin then flows with the bile into the gallbladder for storage and then into the small intestine when you eat.
And what happens to it in the intestine? Does it just stay yellow?
No, the bacteria in your gut get to work on it. Intestinal bacteria convert the conjugated bilirubin into a series of colorless compounds, collectively called urobilinogen.
Urobilinogen, colorless.
Right. Most of this urobilinogen continues down the intestine and is further converted by bacteria into stercobilinogen. Stercobilinogen is then oxidized to stercobilinin.
Stercobilin, is that?
That's the main pigment that gives feces their characteristic brown color.
Ah, so the color comes from broken down hair meat.
Ultimately, yes. Now, a small fraction of that colorless urobilinogen in the gut doesn't get converted to stercobilin. Instead, it gets reabsorbed back into the bloodstream.
Reabsorbed, where does it go?
Most of it goes back to the liver via the portal vein, the enterohepatic circulation. But a tiny amount escapes the liver and gets filtered out by the kidneys into the urine. In the urine, this urobilinogen gets oxidized to urobilin, which is yellow.
So urobilin is what makes urine yellow.
It contributes significantly to the yellow color, yes. So heme breakdown products color both your stool and your urine. Hashtag 4.2 clinical jaundice, icterus. Now, if anything goes wrong anywhere along this complicated pathway from excessive heme breakdown to problems with liver uptake, conjugation, or excretion into bile, bilirubin can build up in the blood.
And when bilirubin levels get high enough, it starts depositing in tissues, especially the skin and the sclera, the whites of the eyes, making them look yellow. Does jaundice, right?
Exactly. Jaundice or icterus isn't a disease itself, but it's a clinical sign that something is wrong with bilirubin metabolism.
The key challenge then for us as students is figuring out where the problem lies based on the clinical picture and especially the lab tests. You mentioned 3 main categories.
Right. We generally classify jaundice into 3 types based on where the primary problem is relative to the liver. 1, prehepatic jaundice, problem before the liver, usually too much bilirubin being produced for the liver to handle. 2, hepatic jaundice, problem within the liver itself. Damaged liver cells can't process bilirubin normally. 3, posthepatic jaundice, problem after the liver, blockage of bile flow out of the liver.
Okay, let's break those down using the lab tests. How do we tell them apart? Starting with prehepatic, which you said is usually due to too much breakdown.
Right. The classic cause is hemolytic jaundice from massive red blood cell destruction, think severe hemolysis after malaria, or sickle cell crisis, or mismatched blood transfusion. The liver is working fine, but it's just overwhelmed with unconjugated bilirubin coming from the spleen.
So the main bilirubin in the blood will be?
Predominantly unconjugated bilirubin.
Okay. And how does that show up on the Vandenberg test?
Unconjugated bilirubin isn't water-soluble, so it won't react directly with the chemical reagent. You need to add alcohol methanol first to make it soluble. So it gives an indirect positive Vandenberg reaction.
Indirect positive. What about liver enzymes like ALT and AST?
Usually normal or maybe only slightly elevated. The liver cells themselves aren't damaged. Alkaline phosphatase, ALP, is also typically normal.
And urine and stool color. If you're breaking down tons of Heme.
You're making tons of bilirubin, which gets converted to lots of urobilinogen in the gut. More gets reabsorbed, so you get dark urine due to high urobilinogen levels. And more stercobilin is made, leading to dark brown feces.
Okay. Prehepatic hemolysis. Unconjugated bili, indirect VDB.
Ah.
Normal enzymes. Dark urine, dark stool. Got it.
Yeah.
Now, hepatic jaundice. Problem in the liver, like hepatitis.
Exactly. Viral hepatitis, toxins like alcohol, cirrhosis, anything that damages the liver cells, hepatocytes. Now the liver has trouble with multiple steps. Taking up bilirubin, conjugating it, and excreting the conjugated bilirubin into bile.
So what kind of bilirubin builds up in the blood?
You can get a mixed picture. Both unconjugated, because uptake and conjugation are impaired, and conjugated, because excretion is impaired. Bilirubin levels rise in the blood.
Mixed. So the Vandenberg reaction would be?
Biphasic. Some conjugated bilirubin reacts immediately, direct, and the rest reacts after adding alcohol, indirect.
Biphasic. And the key lab markers here are the liver enzymes.
Absolutely. Because the liver cells are damaged and dying, they leak their enzymes into the blood. So you see significantly elevated ALT and AST transaminases, often hundreds or even thousands of units per liter. That's the hallmark of hepatic jaundice. ALP might be slightly elevated, but usually not dramatically.
Okay. Hepatic liver damage, mixed bili biphasic VDP, high ALTAST, variable urine stool, makes sense. Finally, post-hepatic or obstructive jaundice.
This is usually caused by a physical blockage outside the liver preventing bile from flowing into the intestine. Think gallstones stuck in the common bile duct or a tumor in the head of the pancreas compressing the duct.
So the liver's working fine. It's conjugating the bilirubin.
Yes, the liver conjugates the bilirubin perfectly well, making it water-soluble, but it can't get out. The bile backs up.
So the bilirubin that accumulates in the blood is
predominantly conjugated bilirubin.
Water-soluble, so the Vandenberg test.
Direct positive. It reacts immediately with the reagent because it's already soluble.
Direct positive. What about the enzymes? ALTAST again?
Not primarily. While ALTAST might be slightly raised due to back pressure, the key enzyme marker for obstruction is alkaline phosphatase, ALP. ALP is found in the cells lining the bile ducts. When the ducts are blocked or inflamed, ALP levels rise dramatically. So highly elevated ALP is the classic sign of obstructive jaundice.
High ALP. Got it. And if the bile can't get into the intestine?
Then the conjugated bilirubin never reaches the gut bacteria. No urobilinogen is formed, and therefore no stercobilin is formed. This leads to the characteristic pale, clay-colored stools.
Pale stools. What about the urine?
Because the conjugated bilirubin backing up in the blood is water-soluble, the kidneys can filter some of it out. This makes the urine dark, but this time it's dark due to the presence of conjugated bilirubin itself, bilirubinuria, not urobilinogen, which is absent or very low because none is being made in the gut.
Okay. Post-hepatic obstruction. Conjugated biliary. Direct VDB. High ALP equals pale stool, dark urine, bilirubinuria. That differentiation is really clear using the labs.
It's incredibly powerful for diagnosis.
Now, briefly, there are also some inherited conditions causing jaundice, right? Not due to hemolysis or obstruction, but problems with conjugation itself.
Yes, genetic defects in bilirubin metabolism can cause jaundice. The most common is neonatal jaundice, or physiologic jaundice of the newborn. Baby livers often have temporarily low levels of the UGT enzyme right after birth, leading to a buildup of unconjugated bilirubin. Usually mild and resolves on its own, but needs monitoring.
Okay. And more serious genetic forms.
There's Crigler-Najjar syndrome. Type I is a complete absence of the UGT enzyme, leading to extremely high unconjugated bilirubin levels and severe neurological damage, kernicters, if not treated aggressively, like with phototherapy or liver transplant. Type 2 is a partial deficiency. Less severe, but still causes significant unconjugated hyperbilirubinemia.
In Gilbert's, I hear that mentioned a lot.
Gilbert syndrome is actually quite common and generally benign. It's due to a mild genetic reduction in UGT activity. People with Gilbert's have slightly elevated unconjugated bilirubin levels, which might fluctuate and cause mild jaundice, especially during illness or stress, but it doesn't typically cause any serious health problems.
So mostly unconjugated bilirubin issues in these genetic types.
Primarily, yes, because the defect is usually in the conjugation step itself.
Okay, let's reinforce that Vandenberg distinction one last time. It seems key.
It is. Remember, conjugated bilirubin is water-soluble, so it reacts directly. Unconjugated bilirubin is insoluble, needs alcohol, so it reacts indirectly. If you remember direct, conjugated, indirect, unconjugated, you're halfway there to differentiating jaundice types.
Excellent. And one final recall question from the heme synthesis part. Which enzyme is rate limiting and which one does lead inhibit at the very end?
Take a second. Rate-limiting enzyme at the start. ALA synthase. Enzyme at the end, inhibited by lead. For chelatase. Knowing those two anchors the pathway clinically. Hashtag, outro. All right, that was a pretty intensive run-through. We covered the elegance of hemoglobin, its structure, oxygen binding, the Bohr effect, 2,5-3-DPG. Then we looked at what happens when things go wrong, sickle cell, Thalassemias. And finally, we traced the entire path from heme breakdown to bilirubin metabolism and how disruptions lead to the different patterns of jaundice.
Yeah, and the big clinical takeaway really is using those biochemical markers. You can literally look at whether the bilirubin is conjugated or unconjugated, check the AL test versus ALP levels, and get a very strong indication of whether you're dealing with hemolysis, liver damage, or bile duct obstruction. It's biochemistry in action, guiding diagnosis.
Absolutely. You've really tackled some core high-yield concepts here. Great job working through that.
Definitely. Okay, before we wrap up, let's leave you with a final thought to chew on, something that connects HB function back to red cell survival itself.
We talked about how 2003 BPG is crucial for efficient oxygen release and how it's made via glycolysis within the red blood cell. Now, consider a patient with severe beta thalassemia. They have that imbalance, unstable alpha chains precipitating, leading to red cell destruction, ineffective erythropoiesis. How might that underlying structural instability and the stress on the red cell itself impact its ability to maintain glycolysis? Could the energy demands of just trying to survive or the damage from precipitated chains compromise ATP production and therefore 2,5-3-BPG synthesis in the mature red cells that do make it into circulation?
So could the thalassemia actually worsen tissue hypoxia, not just by causing anemia, but also by impairing the oxygen release mechanism in the surviving cells?
Exactly. It's thinking about the downstream energetic consequences for the red cell when its primary protein is fundamentally flawed, something to ponder.